Well, hello everybody. Welcome to the week 2 review video. We're getting into the good stuff now, some protein structure, as well as the very beginning of thermodynamics and determining the rate of reactions. This is going to set the stage for what we do next week when we get into Michaelis-Menten enzyme kinetics, which is a wonderful unit of the course, but we're building foundation this week, which is also critically important. So the topics that have emerged as confusing from your review videos, not review videos, from your lecture videos in Edpuzzle, are the ones that you see in front of you. Protein structure, and we're going to review everything all the way from primary to quaternary structure. Order of reactions, specifically this idea that you can be a second order reaction, but first order from the perspective of one of the reactants. We'll try to clarify that a bit. We'll talk briefly about the transition state and how that factors into the induced fit model for enzyme substrate binding, and we'll finish up with a very quick review of free energy diagrams, and this whole notion of deltaG and what that means. So a little bit on our plate, but we'll try to keep it short and brief. So let's get started. First thing you see in front of you here is a wire. This is just a regular wire that I took out of some excess coaxial cable I had lying around in my basement. And we're going to use this to diagram a protein folding up all the way from primary structure to ultimately to quaternary structure, although we can't do that with just one wire, as you'll see in a second. What I have here in blue dry erase marker, which is actually pretty stupid because it's going to come off on my finger as soon as I start touching it, but what we have in blue are peptide bonds. So you can think of each white gap as an amino acid, and the peptide bonds in blue are holding that chain of amino acids together. So this is our primary structure. It's just a single chain of amino acids. It has no form other than being that chain. Now let's move up to secondary structure. Secondary structure is defined as regions of local folding or order. That's what secondary structure is, regions of local folding. And also secondary structures are held together primarily, if not exclusively, by hydrogen bonds. We have three main categories of secondary structures that we discuss in this course: beta sheets, alpha helices, and turns or loops. And we'll diagram all of those in just a second. So let's move from primary structure to secondary structure. Let's induce some local regions of order. I'm going to take this wire, and there goes those peptide bonds. I'm going to loop it around my finger, so there's a region of local order. There's an alpha helix. Now just that region has been ordered or has folded. And what's holding this alpha helix together, the reason why this ribbon of amino acids is holding here, is because we have hydrogen bonds down the axis of the alpha helix. So there's a hydrogen bond from this amino acid to this amino acid, a hydrogen bond from this amino acid to this amino acid. Those hydrogen bonds are actually in the backbone of the protein. They don't involve the side chains. So we're holding this alpha helix in its shape by these intramolecular H bonds within the single chain of amino acids. Let's continue inducing some regions of local order. Let's make a beta sheet over here. I'll do that, and I'll do that. Make it a little bit more crunched. And there's a beta sheet. That beta sheet is made up of 3 beta strands. So we have the ribbon of amino acids is actually doing this zigzag or switchbacking, as I say in the lecture video, holding this into a beta sheet. You can see it's very different than the alpha helix. The alpha helix is a coil. It's 3-dimensional. The beta sheet's pretty flat. It is a sheet. It's mostly 2-dimensional in that sense, but it's also being held together by intramolecular H bonds. There are hydrogen bonds from amino acids here, connecting to here, from here to here, again along the backbone between the amino and carboxy groups. Those H bonds are holding this beta sheet together. So we have 2 secondary structures here so far. We have an alpha helix and a beta sheet. Notice how we took the long wire in its primary form, its primary structure, and we induced local regions of order. We just did local folding. Over here on the left, we folded this into an alpha helix, and on the right, we folded this into a beta sheet. That's our secondary structure. Now let's go to tertiary structure. Tertiary structure is greater folding to create a complete 3-dimensional shape in the protein. So that might be the tertiary structure of this polypeptide. You see, I've introduced our third type of secondary structure. I had to put a turn in here. This is not an alpha helix. This is not a beta sheet. This is a turn holding these 2 secondary structures in their 3-dimensional shape. Now the alpha helix is immediately adjacent to the beta sheet, and it's because of this turn, that additional fold, that made that possible. Now, what's holding this tertiary shape in place is still hydrogen bonds, perhaps. We can also have ionic interactions, hydrophobic interactions. We can have a number of different non-covalent forces. But if we do have hydrogen bonds holding this into its tertiary shape, that's now hydrogen bonds between the side chains of these amino acids, not in the peptide backbone, not between the carboxy and the amino groups. So we went from the long wire in its primary shape to secondary structures, local regions of folding, and then with additional folding we can bring this into a three-dimensional shape or a tertiary structure where we have additional intramolecular non-covalent forces. So this is a fully folded polypeptide. This is in its tertiary shape. 3 levels of structure. The chain of amino acids, local regions of secondary structure, alpha helices, beta sheets, and then turns can bring those together into a three-dimensional shape. For some proteins, not many, but for some proteins, the folding is now done. This protein can go off in the cell and do its job and be done in the tertiary form. But what's much more common is that we go to quaternary structure. What I have here is a second wire that I put into 2 alpha helices before. So if I were to ask you on an exam or a quiz what structures are represented here, you would say primary, because we have a chain of amino acids, and secondary, because we have regions of local order. We have an alpha helix over here and an alpha helix over here. There are hydrogen bonds between the backbone, the peptide backbone of this chain of amino acids, holding these alpha helices together, but I don't see any turns, I don't see any loops, and I don't see any tertiary structure. So this is representing, all we see here is primary and secondary structure. Let me induce a turn to bring this into a tertiary shape. Now we have a tertiary structure. We have a turn or a loop over here, and it's bringing these 2 alpha helices in close proximity to each other. Now the side chains of these 2 alpha helices can interact and hold on to each other through non-covalent forces, keeping this three-dimensional shape. If these 2 polypeptides were to come together and hold on to each other through non-covalent forces, maybe there's a negative charge right here and it's matched with a positive charge here. Maybe this is hydrophobic and this is hydrophobic, and these come together to exclude water. If 2 different polypeptides come together, we are now in a quaternary form. In fact, if 2 or more polypeptides come together, we are on quaternary form. Here's another polypeptide, this one in black. It's made up of an alpha helix and a beta sheet, just like the white one, and it's joining the party here. So quaternary structure are multiple polypeptides coming together to create a larger, and here's where I hate the terminology, to create a larger protein. This whole thing is called a protein, made up of 3 distinct polypeptide chains, but this is the protein. But this protein would also be called a complex, and that complex is made up of 3 subunits: copper, white, and black. So this is a subunit, this is a subunit, this is a subunit of the larger complex, and those subunits are held together by non-covalent forces. This complex will go off and do its job. Most of the enzymes, most of the proteins, in fact, that we see in cellular biology are complexes. They are aggregations of multiple subunits. So that's protein structure. Hopefully we've answered a great deal of confusion by putting it all into context in this way, so you can see it with models. Feel free to get a twist tie and just kind of do this on your own as practice. Primary structure, just the chain of amino acids, a long ribbon of amino acids held together by peptide bonds. Now take that chain or that ribbon of amino acids and introduce local regions of order. An alpha helix, a beta sheet. Now you're in secondary structure. Use additional secondary structures to induce turns to bring those secondary structures into a 3-dimensional shape. You've just reached tertiary structure. H bonds between the backbone of these amino acids hold secondary structures together, and H bonds and additional non-covalent forces hold tertiary structures together. You can stop there for some small subset of proteins, but most proteins have those individual, fully folded, tertiary polypeptides come together to create a larger single protein complex made up of multiple subunits held together by additional non-covalent forces. Whew! That's awesome. And that's protein structure. Okay, so orders of reaction. Now, some reactions, not many, but some reactions are very simplistic. They are first-order reactions. One thing becomes one thing. One thing becomes two things. First-order reactions. What impacts the rate of this reaction? Well, pretty much just the concentration of A. The more A you have, the more B you should make, right? So that's what a first-order reaction is. Most reactions that we encounter are not first-order. They tend to be second order or more. In second order reactions, two things come together to create some product. We have two reactant molecules, A and B, and perhaps here they're being joined to make C, or maybe they're modified to make C and D. These are your products. This is a second order reaction because two different things can impact the rate of the reaction. The concentration of A or the concentration of B. Increase the concentrations of A or B, you should get a faster reaction. Increase the concentration of both, second order, you're going to get a faster reaction, second order reaction. But what seemed to give a number of you some trouble, and honestly for good reason, because it's a pretty abstract concept and it's not the most intuitive, is you can still have first order reactions within a reaction like this. If you begin talking about the reaction from the perspective of one of the reactants. Okay, what do we mean? What we mean is, let's assume that you're B. You are the molecule B, and your concentration is fixed. We can't add more of you, we can't add less of you. And you're looking at this reaction from your perspective. From your perspective, how many things can be changed to increase or decrease the rate of this reaction? Well, your concentration is fixed, and you can't really look in a molecular mirror. So from your perspective, the only thing that can be changed in order to increase or decrease the rate of this reaction is A. Add more A, you'd expect the reaction to go faster. Add less A, the reaction will slow down. So from your perspective as B, this is a first-order reaction. Only one thing can be modified or changed in order to make the rate of the reaction alter. And you can flip that. Now, let's say that you are A, and you're looking at this reaction from your perspective. Your concentration is fixed, and you can't look in a molecular mirror. From your perspective, this is a first-order reaction as well, because the only thing that can be modified from the perspective of A, from the perspective of you, the only thing that can be modified to alter the rate of this reaction is the concentration of B. So from the perspective of A, this is a first-order reaction. Why do we introduce that? Well, biochemists use this to simplify complex reactions and measure their rates. And next week, we're going to be going into some of the weeds of rates of reactions as we talk about enzyme kinetics. We won't really mess too much more with first and second order reactions per se, but it's important for you to know those definitions as we use them in some of our discussions later on in the course, especially next week and the week after. So, order of reactions basically gets at the notion of how many things can be changed or modified in order to change the rate of a reaction. Second-order reactions tend to be fairly common in biochemical systems. That means two things can be altered to impact or change the rate of the reaction, but that can get complicated in a hurry. So what we do is we fix the concentration of one and say, now this is a first-order reaction because only B can impact the change. Or we fix the concentration of the other and we say, now this is a first-order reaction. We can only change A. So really that's all we were trying to get at there. Okie dokie. 15 minutes. Look at us. We're rock stars here. All right. Now, on to transition states and induced fit. Let's talk about transition states first. We kind of go the other way in the videos. Maybe it'll hammer this home if we go in the reverse direction relative to how we first introduced these concepts in the lecture videos. So a transition state is something that is not reactant anymore. Let's go back to a second order reaction, but let's say that A and B come together to make C. So this is going to be a joining reaction, right? We're going to take these two independent molecules and covalently join them into a new third molecule. So we're going to take A and B and make it C. In other words, it's like making a dipeptide. We're going to take amino acid A, amino acid B, and put a peptide bond between them to make them a single dipeptide molecule. This is a second-order reaction. We have two things that we can alter. Now, on the journey of this joining, on the journey of making this peptide bond in this example, as we go from A independently and B independently to C, we're going to go through a transition state. The transition state is no longer reactant, right? This kind of hybrid structure where A still has a bit of its own identity, B still has a bit of its own identity, but they are now covalently linked. They're not quite C, but they're no longer independently A and B. That's your transition state. There's no other way to really describe it other than saying transition states have residual identity to what they were when you started and are not quite what you want to be when you're done. They are a middling, a high energy state between reactant and product. That's your transition state. The analogy that seemed to resonate with quite a bit of you from the lecture was this idea of making bread, right? If the flour and the water and the salt and the yeast, if those are your reactants and the bread, the loaf of bread, is your product, obviously you need to do a little bit of work to get from those ingredients to a fully cooked loaf of bread. You have to mix the ingredients, knead the dough, let the dough rise. So that bread dough, that's your transition state. It certainly is no longer reactant, but it's not quite a loaf of bread yet. It's a middling point on your journey from going from reactants to product. So that's your transition state. Again, this is a very high energy molecule. What do we mean by high energy thermodynamically? We mean it's very, very unstable. You need to put work, effort, energy into the system in order to make the transition state. That's going to become important as we go into this next topic. Making that transition state is the hardest thing to do in a chemical reaction or a biochemical reaction. Once the transition state is made, you're home free from an energetic perspective. So making transition state is key to getting any reaction to proceed forward. That's why we're going to talk about induced fit right now. So here's an enzyme. And that enzyme has an active site. The active site of the enzyme is what binds to substrate. Now, what you've been taught before is the lock and key analogy, right? And my apologies on behalf of all former instructors. Many times these lies are a necessity in order to keep things simple. If we try to explain everything to you from the very beginning how it actually works, it would be completely overwhelming. So we tell these white lies along the way. For those of you that had me in genetics, for the whole first half of genetics, I tell you that hair color is controlled by a single gene. Brown hair is dominant to blonde. Eye color is controlled by a single gene. Brown eyes are dominant to blue. And then about halfway through the course, I apologize to you all and say I've been lying to you for the last 8 weeks because there's no such thing as a single gene for hair color, a single gene for eye color. We use that as an oversimplification. Same here. Lock and key is somewhat valid in the sense that only specific substrates will bind to an enzyme, but it's not this perfect fit kind of thing that you think of with a lock and a key. The protein is malleable, the enzyme is malleable, it's flexible, and it accepts substrate like a lock can accept a key, but the protein, the enzyme, actually forms around and manipulates that substrate, something a lock would never do to a key. But this induced fit model is the more accurate model, and it is important for talking about transition state, which we'll put into context in just a second. So as a side note, though, I had this conversation with one of you over Canvas. It does make you think, with all these lies that you've been told and you're finding out about now, it must make you worry if you take a moment to reflect on what additional lies have you been told that haven't been admitted yet. And there are many oversimplification lies that you will continue to be told as your undergraduates. If you want to know the real truth, get a PhD, I suppose. So let's go back to our example as A and B coming together to create C. So A fits into the active site here, let's see, and B fits into the active site here, let's say, as well. Now that's our first step. That's enzyme binding to substrate. But then what happens is the enzyme, it had to kind of move. You can even see I didn't quite draw it properly, right? Look, I've got A overlapping with green here, and that can't be. This can only take one physical space. So the enzyme moves and it flexes to accommodate the binding of substrate. That's that induced fit. The enzyme kind of molds around substrate, but the enzyme does not like moving and molding. The enzyme would rather go back to its initial form. So once substrate is bound, once you have enzyme and substrate together, the enzyme recoils and it goes back to the form that it's most comfortable in. But in doing that, it's applying pressure on the substrate. So as this enzyme recoils back to the shape that I drew before, what we see is that we're now pushing A and B together. Ooh, we're pushing A and B together. In other words, what the enzyme is actually creating is transition state, and that's the secret. That's the secret you're going to hear fully next week, but I might as well divulge it now. The secret to enzyme activity is forcing transition state to occur. What the lock fits perfectly, the key to the lock of any enzyme is transition state. What the enzyme wants to bind to is transition state. What the enzyme winds up creating is transition state. Substrate binds to the enzyme because it's pretty damn close to transition state, and the enzyme will accommodate it. But it causes the enzyme to deform. It's not energetically favorable. So when the enzyme recoils back to its preferred structure, it squeezes and pushes substrate into transition state. And once you've made the dough, baking the bread, you're home free. Once you've made transition state, the rest of the reaction proceeds on its own. So enzymes make transition states. The induced fit model states that the enzyme can accommodate the substrate, but it has to deform and mold around substrate to bind it properly. The enzyme is not happy doing that, and as soon as it's given the opportunity, it recoils to its preferred shape, and that squeezes reactants together into transition state. So the induced fit model is directly relevant to transition state. The reason why we don't bind to substrate perfectly, the reason why we don't bind to substrate as though it was a key in our lock, is because that would be the most favorable binding around. Enzyme would bind to substrate and say, wow, this feels good. This is a perfect fit. Hand in glove. Met my match. Why the hell would I want to change that? If I'm the enzyme, why would I want to alter substrate if it fits me perfectly? No, instead, substrate is an almost perfect match for enzyme. It's almost the perfect partner. I love that sense of humor. I love that eye color, but I really wish they'd stop complaining about the toothpaste. I will induce my fit and accommodate the weaknesses of this substrate right now, but then as the enzyme, I'm going to work and work and work on that toothpaste idiosyncrasy. I'm going to push and stretch and deform this substrate into transition state because that's what I want to bind to. That's what makes me happiest. Enzymes force substrate into transition state. And then what? Well, the then what gets to changes in free energy. So we could say that this is time down here for now, and this is energy here on this axis. And we can put the energy of the reactants maybe here, and the energy of the products maybe here. So here's the reactants, and here's the products. And this is the course of the reaction over time. We start with the reactants that contain some amount of energy. Now, what do we mean by contain an energy? Maybe we should take a step back there. Everything contains energy. That was one of Einstein's main contributions to our science. The whole premise of the atomic bomb is that everything contains energy inherently, and we can, under certain conditions, release that energy. The covalent bonds of all molecules contain energy. I mean, that's the premise of ATP. ATP is the battery of life because there's high energy in that third phosphate bond. All molecules contain energy. So if you've got a pile of reactants, and those reactants are molecules, there is inherent energy stored within the covalent bonds of those reactants. And then if you have a pile of product, there's an amount of energy contained in that product. you'll have differences in those energies. Sometimes the sum total of all the energy contained in the covalent bonds of the reactants is greater than the sum total of all the energy contained in the products. And as that reaction goes forward, if you have more energy in your reactants and less energy in your products, you're going to have an energy differential. And that energy is released to the environment. Remember, less energy in the products, more energy in the reactants. That energy differential is released to the environment as heat. Heat makes molecules move faster, boiling water. Molecules moving faster are more random, entropy, and the universe loves entropy. Favorable reaction, spontaneous reaction. This is a reaction that will occur. But you can consider another case where all of the sum total of energy inherent in the bonds of the reactants is less than all of the energy in the products. You still have an energy differential, but now you actually needed to take energy from the environment and put it into products. In order to go from the energy in the reactants to the energy of products, you needed more energy. You needed an energy gain. And that meant energy had to go into this reaction. You stole heat from the universe. You made the universe colder with this reaction. Colder things move more slowly, ice. Slow-moving things are less random, entropy, and the universe hates to lose entropy. Not a spontaneous reaction. Universe hates this. You want this to go forward, you got to pull some biochemical tricks, and we'll talk about that in a number of weeks. So favorable, unfavorable. Energy released, energy needed. That's the premise of thermodynamics, at least as far as we need to consider it. So if we use the term G for energy and we use the term deltaG for a change of energy, we can have a pretty simple formula in our back pocket, which is the change in free energy of any reaction that we will consider is the energy of the products minus the energy of the reactants. If we look over here, what we can see is that when this number is bigger than this one, as we've drawn it here, when this number is bigger than this one, 4 minus 8 is a negative value. So negative deltaGs are favorable reactions. They're spontaneous. They release energy. If we go up here and we say, well, this is 8 and this is 4, well, 8 minus 4, that's a positive deltaG. Positive deltaGs require energy. They are not favorable. So we just tied this idea of free energy to deltaGs to the spontaneousness or favorableness of reactions. Reactions that release energy, release heat, they make the universe happy by increasing entropy. Those reactions have a negative deltaG and they are spontaneous. Conversely, reactions that need energy, need heat, they have more energy in their products than their reactants, they're going to have a positive deltaG and be non-spontaneous because the universe doesn't want to be colder. It doesn't want to be less random. Last point there, and perhaps the most important to set us up for next week. What we've been talking about here has been path independent. We magically went from reactant to product. But we don't attribute that to magic anymore. We know what lies between reactant and product. Transition state does. We have to make the dough. And so, if we need to make transition state, that means we need energy. We need to squeeze transition state to make it. We need to put energy into transition state. I told you that transition state is a high energy molecule. Well, there it is, high energy, right there on the free energy diagram. Making transition state is the barrier to any biochemical reaction because transition state is unfavorable, unstable. There is more energy in the bonds of the transition state than either reactants or products alone. That cools the universe down, and that makes the universe unhappy. Now, the universe has a potential net gain of free energy if it's just willing to invest in the transition state and put that energy in temporarily. It's going to get all that energy back plus profit. Is the universe willing to do that? Usually not. Do we know of anything that wants to make transition state? Do we know of anything out there that really, really likes transition state? Anything that will actually recoil and put its own energy in to create transition state? Of course we do. Enzymes' entire job is to make transition state. Enzymes want to bind to transition state. Enzymes are made to bind to transition state. Enzymes will bind to substrate and then squeeze it and pull it and squish it and move it and manhandle it, or womanhandle it, in order to create transition state. Enzymes do this work for us. In fact, enzymes, because of their inherent fondness for the transition state, actually lower the amount of energy needed to make transition state. And the universe is willing to make this kind of investment even if it wouldn't have made this one. So enzymes lower the energy required to make transition state, and in doing so, enzymes smooth the bump needed to get from reactant to product. That is definitely stepping on the toes of Michaelis-Menten, laying the groundwork of what we're going to talk about next week. If that is not entirely clear to you, no worries, because again, we're going to be spending a lot of time on this material next week. But the whole point of introducing these concepts right now is to get you comfortable with this way of thinking, and so it made complete sense, I think, to bridge week 2 to week 3 by letting you peek behind the curtain and see a little bit of these energetics a little bit more deeply. So hopefully these 4 concepts are clearer than they were before. This covers the vast majority of what almost every single one of you said was confusing. But please do remember that I'm giving each of you personalized and individualized feedback in Edpuzzle. So check Edpuzzle, go back to those assignments, and take another peek. Because if I didn't answer your question here, there's a very, very high likelihood that I answered you specifically in Edpuzzle. Thanks so much for watching, and I guess I'll catch you all when we're recapping this week's challenge questions.